Have you wondered about how plants use sunlight energy and convert into chemical energy during photosynthesis? How does that happen? We know it occurs in a chloroplast, but what actually happens? Well, I can tell you there are molecules in the plant called pigments, and these molecules absorb the sunlight energy and convert it into chemical energy in photosynthesis. There are two types of pigments, chlorophyll A and chlorophyll B. These molecules are similar in structure, except that chlorophyll A contains a methyl group and chlorophyll B contains an aldehyde group. This slight change in structure allows them to absorb different kinds of light. So chlorophyll A likes to absorb blue-violet light and chlorophyll B likes to absorb red light. What they don't like to absorb is green light, and that is what they emit. So that's why whenever we look at plants, we see green. Now, how about in the fall? The leaves change color from green to red, orange, and yellow. How does that happen? Didn't I just say chlorophyll A and B emit a green color? I think it's time to go back to my lab and conduct an experiment. So today we're going to be analyzing the plant pigments using paper chromatography. So in order to start this experiment, you will need one clear cup, a smallish pencil, a small coin, some scissors for cutting, a small ruler, any sort of green plant. Today we're going to be using the leaves from a celery stick, some tape, some isopropyl or rubbing alcohol. I'm going to be using 70%, but you can use 90% as well. You're going to need some coffee filter papers, and finally, most importantly, do not forget your safety goggles. So to begin, use your coffee filter paper and cut a strip that is 1 inch wide and 2 inches tall. I've already done that. It should look something like this. Then you get your pencil and your ruler and you draw a straight line at the bottom of your filter paper about 1 centimeter up. Then you want to rip a leaf from your plant and use your small coin. So the leaf has a shiny part and a non-shiny part. So look at the shiny surface and put that outwards and put the coin inside and wrap the leaf around your coin. And then use this against a hard surface to get those green plant pigments along that line you just drew. So when you do that, you should just be making sure the green pigments are everywhere along that line. If your leaf's not giving you enough pigments, just switch the position of the coin inside that leaf and keep going until you have a green line drawn. Okay, so it should look something like this with that green line drawn here. Next, you want to get your tape and you want to tape it on your paper onto your pencil, and you want this filter paper to be straight. So first I like putting the tape on the filter paper and then putting that on this pencil. Okay, so now you should have something like this, your pencil and your filter paper hanging down with your green line at the bottom here. So next you want to put your rubbing alcohol into your clear cup, and supporting your cup is clear so that you can see through it. So I'm going to put about 30 milliliters of rubbing alcohol into the cup. And then you want to get your pencil and slowly lower the filter paper into the cup. So you want to make sure your filter paper is touching the surface, but also make sure that green line you just made is above the surface. So if we take a look here, it looks good. And now we want to wait 10 minutes and see what happens. 10 minutes has passed, and now we will be removing the filter paper from the cup and putting it on a clean white paper towel. As we can see here, there is the green pigment that we put on the filter paper, but now there is a yellow color above it. So why did that happen? Why did we see a yellow color above the green color when we just put green pigment on the filter paper? Well, chromatography is actually used to separate the different parts of a plant pigment. Do you remember how I said chlorophyll A and B have the same structure? Well, there are other pigments in a plant called carotenoids that have a totally different structure than chlorophyll A and B. And during this chromatography process, they actually got separated. Carotenoids are present alongside chlorophyll A and B in a plant, but they actually only become active in the fall when chlorophyll A and B no longer work. They help in photosynthesis and they also help maintain the plant's structure. Do you remember how I said chlorophyll A and B emit a green color? Carotenoids actually emit a red, orange, and yellow color, and that is exactly what we saw here today. So now when you're walking in the fall and you see the leaves changing color, you will know exactly what is happening in that plant on the microscopic level. I hope you enjoyed doing this experiment with me, guys. Thanks for watching. Bye!